Hello, APBIO students. This time we are going to look through Chapter 7 notes, which covers the cell membrane structure and function, as well as specifics on cell transport. We will cover the plasma membrane, membrane permeability, membrane transport, facilitated diffusion, tonicity and osmoregulation, and mechanisms of transport. Let's start with the specifics of the cell membrane. So the cell membrane is a fluid mosaic of lipids and proteins. Phospholipids are the most abundant component, and they have hydrophilic and hydrophobic regions. The heads of a phospholipid are hydrophilic, and the tails are hydrophobic, which is why they arrange as they do in water, with the hydrophilic regions on the outside, with the hydrophobic regions trapped in the middle. The fluid mosaic model of the cell membrane tells us that it is a mosaic of protein molecules bobbing in a fluid bilayer of phospholipids. So cell membranes are not rigid, they are fluid, they are moving all of the time. Here is a cool looking diagram of the cell membrane where you can see the phospholipids, some giant proteins, glycoproteins and glycolipids for cell recognition, and lots of other things that might be there to help with cell functions. Proteins in the membrane will also have hydrophilic and hydrophobic regions depending on where they are and what they do. They are not randomly distributed in the membrane, but they are grouped in long-lasting specialized patches. So like all the things that let one type of thing in or out of the cell will be kind of clumped together in a region. As I said, membranes are fluid. They are not rigid. They're held together mainly by weak hydrophobic interactions. Most lipids and some proteins will shift sideways, and it remains fluid unless temperature decreases too much. So if you think about bacon grease cooling. How fluid it is also depends on cholesterol that is bound within the membrane to help with fluidity. And the fluidity of the membrane affects permeability, which is like how easy it is for something to get in and out, and the ability of proteins to move where they are needed. So you can see in picture B here that we have cholesterol in our animal cell membrane that helps with fluidity. The cell membrane lipid composition varies between species, and there appear to be adaptations to maintain appropriate membrane fluidity under specific environmental conditions. For example, fish that live in cold environments have membranes with a high proportion of unsaturated hydrocarbon tails. And in some organism, the proportion of components will change during the warm and cold seasons. So depending on where the organism lives, their lipids may be slightly different. Phospholipids, while they are the main component of the membrane, proteins determine the function. Each cell will have a unique set of proteins, and there are two main types, integral and peripheral. Integral proteins will penetrate the hydrophobic interior of the bilayer. They can either go partway through the membrane or go all the way through, and they will have hydrophobic and hydrophilic regions. Peripheral proteins are loosely bound to the surface of the membrane only. So here are some functions of membrane proteins. The first picture will move from left to right. We have some energy that's helping whatever this thing is move out the cell. We have enzymes that help speed up chemical reactions. We have a signaling molecule with its protein receptor, which we'll talk about a lot more in Unit 4. And then we have a cell recognition example here on the far right of a glycoprotein that is, letting the cell next door to it, hey, it's cool, I'm supposed to be here. Let's talk about the role of membrane carbohydrates in cell-to-cell recognition. This is crucial to an organism's functioning. Sorting cells into tissues and organs during development, we need that for that. And it is the basis for rejection of foreign cells by immune systems. And so that's part of the reason why people who maybe have to have an organ transplant can have issues, because at some point their body may say like, hey man, like this is not supposed to be here, we have to get rid of it, which can lead to organ failure and rejection. Cells recognize cells by binding to molecules on the surface of the plasma membrane. The structure of these carbohydrates for cell recognition are short and branched. They can be glycolipids, which is a sugar bonded to a lipid, or glycoproteins, where a sugar is bonded to a protein. And these will vary between and among species. So even though we're all human, ours do not all look exactly the same, and that's okay. Moving on to the permeability of the bilayer. So the membrane is selectively permeable, which means that only some things can go in and out. Nonpolar molecules like hydrocarbons, CO2 and O2 are hydrophobic or nothing, and they can cross the membrane freely, meaning they just squiggle their way through the phospholipids. Ions and polar molecules must use a protein to cross the membrane. So we have transport proteins that go all the way through the membrane, which help the ions and polar molecules cross. Some proteins that are called channel proteins are like a hydrophilic tunnel through the membrane. For example, aquaporins help water get across. Water cannot go through the membrane all by itself. It needs a channel protein for it to be able to go through. And some carrier proteins will change shape to help the substance cross. So we will talk about 3 types of transport: passive transport, active transport, and bulk transport. Starting with passive. Passive is the diffusion of a substance across the membrane without using energy. So diffusion is the movement of particles of any substance so they can spread out. And they will move, wait a second. That is incorrect. They diffuse from an area of high concentration to an area of low concentration, meaning it goes down the concentration gradient. So we don't want a huge amount of O2 on the outside of the cell. It will force its way through the membrane to go to the inside of the cell in search of balance. When we talk about a concentration gradient, that is the region along which the density of a substance increases or decreases. So we have a gradient when there's lots of O2 on one side and not a whole lot on the other. So here is an example of diffusion here, which you can see the molecules and the arrows in which they are moving. Osmosis is the facilitated diffusion of water. So solutions with higher solute concentration have a lower water concentration. So water diffuses across the membrane from an area of high water concentration to low water concentration to balance the solute concentration. So we can see that from the famous YouTube experiment. If I have a U-shaped tube with a semi-permeable membrane down the middle, and I have a higher concentration of solute on one side, water will move so that it's, higher on one side of the U than the other, so that the concentration of the solute will be closer to equal. Let's talk about water balance of cells that don't have a cell wall. So we're thinking about animal cells here. Tonicity is the ability of a solution surrounding a cell to cause that cell to gain or use water. And that will depend on the concentration of solutes that cannot cross the membrane. So let's think about an animal cell placed in 3 different solutions. If the solution is isotonic to the cell, we have no net movement of water across the membrane. So it's moving in and out at the same rate because the concentration of things on the inside of the cell and on the outside of the cell is the same. If the solution is hypertonic to the cell, we have a net movement of water out of the cell, which will cause the cell to shrink and probably die. If the solution is hypotonic to the cell, meaning it will have a lower water concentration and more solutes on the inside, we have a net movement of water into the cell, which causes the cell to swell and burst. Cells that don't have a cell wall cannot tolerate excessive increase or decreases in water. And so there are several adaptations for osmoregulation of organisms. For example, paramecium, which are a type of aquatic organism usually found in ponds, have a contractile vacuole that pumps out excess water to keep too much water from building up on the inside of the paramecium, which could cause it to burst and therefore die. Let's look at water balance in cells with cell walls. The cell walls help maintain the cell's water balance because a cell wall can only expand so much before it puts pressure back on the cell, which is called turgor pressure. When we have that pressure coming back, the cell becomes turgid, which is a good thing for most plant cells because we want them to be sturdy and healthy and happy. When those cells get in a hypertonic environment, they will lose lots of water and shrivel, and then the plasma membrane pulls away from the cell wall, and that is what causes our plants to wilt. So we do want some turgor pressure to keep the plant upright and happy. So here is an example of an animal cell and a plant cell in those three solutions that we just described. So it's hypotonic. For animals, that's a bad thing because a cell can burst. For plant cells, that's a good thing. Normal, in and out at the same rate. And then the last one in a hypertonic solution, shriveled, or in the case of the plant cell, pulled away from the cell wall, which is not good for plants or animals. In facilitated diffusion, which is a type of passive transport, proteins are there to help that molecule move in or out. But there is no energy. The transport proteins will be specific for what they allow in and out. And again, there are two types of transport proteins, channel and carrier. So our channel protein is literally just an opening for the whatever that thing is to move in and out. And then carrier proteins kind of open and close as needed to move things in and out of the cell. If a channel protein transport ions, it's called an ion channel. And many are gated, so they open and close in response to a stimulus, like an electrical impulse for nerve cells or by something binding to the channel. But again, that does not require any energy for it to work. Carrier proteins undergo a shape change to translocate the substance across the membrane, which is the case with how glucose moves in and out of the cell. Moving on to active transport. Active transport does require energy because we're moving things against their gradient. So we're moving something from low concentration to high concentration. It requires carrier proteins, and an example here would be the sodium-potassium pump. So animal cells will have more potassium than sodium on the inside of the cell because of how that pump works. And ATP hydrolysis, so breaking off that third phosphate group, supplies the energy for active transport. So here is an example of the sodium and potassium pump. So what happens is sodium that is on the inside of the cell will attach to the protein, and then ATP binds, which causes that flip in the shape. And so then sodium is released to the outside. Then potassium is able to bind to that same protein, and then it gets pumped into the cell. And so it's all the time pumping sodium out, potassium in, back and forth using energy. Let's look at how ion pumps maintain membrane potential. So membrane potential is the voltage across a membrane. All cells have voltages, which are electrical potential energy, across their plasma membranes. The cytoplasmic side of the membrane is negative in charge relative to the extracellular side, and membrane potential favors passive transport of cations into the cell and anions out of the cell. Last, we are going to look at bulk transport, which will be endocytosis and exocytosis. So large molecules like proteins and polysaccharides and other larger particles cross the membrane in bulk packaged in vesicles using energy. So during exocytosis, we have the secretion of molecules by the fusion of vesicles containing them with the plasma membrane. So the transport vesicles bud from the Golgi, move along microtubules until they run into the plasma membrane. And so then the vesicle fuses to the plasma membrane, and the plasma membrane expands to contain the vesicle. And those things get released into the outside. So insulin and neurotransmitters are secreted that way. With endocytosis, we have the uptake of molecules and other particulate matter via vesicle formation from the plasma membrane. So what will happen is a small area of the plasma membrane will sink in and it forms a pocket, which will pinch until there's a vesicle around that thing trying to get into the cell. And there are 3 types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. And here are examples of that here. With phagocytosis, it's like, an amoeba eating a large food particle. You can see it pushes its way through the membrane until it forms a vesicle around that food particle. With pinocytosis, we have different solutes that, get pinched in and pulled in. And then with receptor-mediated endocytosis, only the things that can bind to the receptor and fit will be brought into the cell. Which you can see those little purple triangles fit perfectly into that Y-shaped receptor. And so those things will, after they're ready, get pinched and pulled into the cell. And that is all you need to know about the cell membrane and transport.